So good morning to everybody. So today I will be talking about the last part of pattern formation, and that will be the anterior posterior axis pattern which is formed in Drosophila. So we had gone slowly on the different types of patterns which are formed and different types of specification, and now we will go to the last specification, that is syncytial specification. We had also studied briefly about what are cytoplasmic determinants present. One thing I would like to mention, that when an egg is formed, especially in the case of insects, the Drosophila, say an Drosophila egg, you have different cells inside this particular egg when it's developing, and this is very different. You have several nuclei present, and there is only inside one plasma membrane you have many nuclei present inside, and such a structure is known as syncytium. And there are certain factors or determinants, cytoplasmic determinants or cytoplasmic factors. They can be mRNA molecules, they can be proteins. Certain of these factors are present in the cytoplasm, and you would expect that inside one egg that these factors will be uniformly distributed throughout the egg. But unfortunately that is not the case. The factors are not uniformly distributed. They are localized in certain areas, and in some areas another factor may be high, and in another area another factor may be high. So since it is not uniform in one area, say maybe in the head region in Drosophila there is a factor protein known as bicoid. The bicoid is much more pronounced in the anterior region, and if it's present in the anterior region, all the cells in the anterior region, they will get transformed into cells which will become the head. And in the posterior region you find another factor known as nanos present. So that will that factor will convert all the cells in that region, in the near the tail region, posterior region, into structures which will make the abdomen and the tail posterior organs. You can't say a tail of a Drosophila, that's a very wrong term, but metaphorically speaking, towards the tail region. So with this, let us start today's class. I will start sharing my screen. Okay, let us start with syncytial specification. Now what is syncytial specification? So a cytoplasm that contains many nuclei is called a syncytium, which I explained right now. And the specification of of presumptive cells within such a syncytium is called syncytial specification. So what happens is that inside the syncytium, where if in former times you had seen different types of cells, and there were certain factors or cytoplasmic determinants which specified the head region, the nerve cell, certain cells to become the nerve or certain cells to become the muscles. That was specification and then differentiation. Here what happens, since the cells are not separated by plasma membrane, you have a whole big cell, inside you have several nuclei present, and this results in a different type of structure. And so even then these nuclei, they specify into certain structures. the anterior part becomes different, the posterior part becomes different. That is because of the presence of different determinants which are not uniformly present throughout the cytoplasm. It may be higher in one region, it may be lower in another region. Similarly, another determinant may be high in one region and it may be low in another region. So, the most important example is that in Drosophila development, where everything is more or less known. During its early cleavage stages, the nuclei divide through 13 cycles in the absence of any cytoplasmic cleavage. Only the nuclei divide 13 cycles, 13 times. There is no cytoplasmic cleavage, that is, the cells are not differentiated, but inside one cytoplasm you have many nuclei present. This division, it creates an embryo with many nuclei contained within one shared cytoplasm, surrounded by one common plasma membrane. This embryo is called the syncytial blastoderm. It is within the syncytial blastoderm that the identity of future cell is established. So, simultaneously across the entire embryo along the anterior to posterior axis. So, the syncytial blastoderm is formed, and then the anterior end, which is the head, and the end of the abdomen, which is the posterior end. So, an identity is established. So, depending upon this, in the syncytial blastoderm, you have head structures, you have middle structures, you have posterior structures are formed in insects. So, let us first have a look at what Drosophila development looks like. So, nuclear cycles during Drosophila early development. So, in the pre-blastoderm stage, cycles 1 to 9. From cycles 4 to 6, these red dots are the nuclei. You have all these red dots, these nuclei are organized like this. Then from cycles 8 and 9, cycle means to division, one cycle is one division. From cycles 8 and 9, nuclear migration towards cortex. These nuclei which are situated center, they migrate towards the cortex side periphery. They start migrating towards the cortex. In the from cycle this was the pre-blastoderm time. Then cycles 8 to 9, nuclear migration towards the cortex. Then from cycles 10 to 13, that is they are known as the syncytial blastoderm cycles. So, there are certain pole cells formed here. These nuclei are migrate over here, and some cells are formed. Inside you have certain of these nuclei present, as well as you can see along the border you have several nuclei arranged. And as the cellular, now in the next step, cellularization starts to take place. They organize the cells in the syncytium, they start to develop certain plasma membrane between the nucleus, and eventually in the cellular blastoderm, which is formed finally, you find individual cells which are differentiated, the nuclei are differentiated from each other. This is in the final stage of development of the particular embryo of Drosophila. But initially at this stage, the syncytial blastoderm stage, these nuclei are not separated by plasma membrane. They are not separated by plasma membrane. It is not present. This occurs in the next following stages when cellularization takes place. So, this is how Drosophila development takes place. So, this is the syncytial blastoderm stage. Keep in mind, all nuclei are arranged in the center and also in the poles. The next thing is opposing axial gradients, they define the position. So, what has emerged from numerous studies is that the cytoplasm of the egg is not uniform. Like I said, that if a factor is present, it is not present uniformly throughout the cell. Some region of the egg has got it much higher, some may have it lower. So, instead what the cell has got, it contains gradient of positional information that dictates cell fate along the egg's anterior to posterior axis. Now, this will be a bit clear when I show the figure. So, in the syncytial blastoderm, nuclei in the anterior part of the cell are exposed to cytoplasmic determination factors that are not present in the posterior part of the cell and vice versa. So, cytoplasmic determination factors present in the anterior part of the egg, they act on nuclei which are present in the anterior part, and they don't act on nuclei which are present in the posterior part. So, the anterior cytoplasmic determinant factors, they convert the anterior part into the head and head and corresponding regions. And so, this is what you mean by positional information. Since the position of the cytoplasmic determination factors are in the anterior end, so they give positional information to the nuclei present in the anterior end, that when you become a cell, then in the cellular stage, then you become cells of the head. So, information is determined here. This is what I meant to say here. So, let us look at the picture. So, this is an important picture from in Gilbert, figure 2.12. So, nuclei positioning and morphogens during syncytial specification. Nuclei positioning and morphogen during nuclear specification in Drosophila melanogaster. So, the nuclei are dynamically ordered within the syncytium of the early embryo, holding their position using the cytoskeletal elements associated with them. So, how do the nuclei move inside the egg or embryo? There are certain cytoskeletons present, and because of these cytoskeletons, some nuclei are on the anterior end and some are go to the posterior end, and their organization is determined by this manner, this factor. So, let us look at this picture D. So, the D is the anterior posterior specification or axis formation. So, you can see this is the egg and these are the nuclei present in the egg. So, in the egg, in this anterior region of the egg, there is a factor known as bicoid in the cytoplasm. This is a morphogen or cytoplasmic determination factor, bicoid. See the pink color region. So, bicoid concentration is much higher over here. In the posterior end, you have caudal present, and because of this caudal is highest in the posterior, and gradually there is a gradient. As we go to the anterior end, this gradually declines. Same with bicoid, as it goes to the posterior end, it declines. So, cells in the anterior end are more exposed to bicoid, and cells in the cytoplasmic end, they are more exposed to caudal. So, these morphogens, and then since there is a gradient, cells over here are intermediate. As you go from anterior to posterior, here the concentration of bicoid is more, concentration of caudal is less. Same thing which happens over here. And so, the inside the nucleus, the nuclear genes are by this gradient, different concentration of this gradient because defines certain nuclear genes in these particular cells. Result that because of this this part becomes anterior, this part becomes posterior, and when cellularization takes place, this region will become the head cells, this will become the thorax cells, where there is a mixture of bicoid and caudal, and then this region there is more caudal over here. So, this region becomes into your abdomen, and there is the so called as such no tail in insects. So this is how the anterior posterior pattern formation takes place inside a Drosophila. With this I end my class, and I will upload this in the Google in the Google Classroom. And I hope you people, if you have problems, you can ask the questions right now. If you have problems, I will answer your questions so far. কারোর কোনো কোয়েশ্চেন আছে? জিজ্ঞেস করতে পারো, হাতটা তুলতে হবে তোমাকে, নয়তো আমি ভিডিওটাও পাঠিয়ে দেবো। আমার তো পাঠিয়ে দিই ভিডিওটা, ইউটিউবের লিঙ্কটা পাঠিয়ে দেবো, তোমরা শুনে তারপরে যদি কিছু না বুঝতে পারো তাহলে আমাকে জিজ্ঞেস করো, হোয়াটসঅ্যাপে লিখে দিতে পারো, ফোন করে জিজ্ঞেস করে নিতে পারো, ঠিক আছে? ওকে, দেন since there is no question, I end my class and I wish you a very happy day today. ভালো থেকো।